Cool. Yeah, thanks for attending the talk, and so sorry for being late. I messed it up completely, so all shame and blame on me. Luckily, it's working out still, I think. Okay, so I gave this talk already, and I just did a little update, but a little update is actually quite impactful, as you will see later. So this was the starting slide from the last lecture I gave, I think 1 or 2 years ago, and the title was Deep Learning in Genomics, and the case for domain agnostic modeling, and I'm stressing and emphasizing on the domain agnostic here. So I have now updated this first deck, and actually I added just one word. Let's see if you can spot it. It's actually automated, and I will come back to that later, why I think we have to automate things. So let's actually have a look. So I'll do a relatively fast but maybe necessary reminder of all the different neural network architecture components, because they are just occurring over and over again, and maybe one or the other isn't familiar with all of those. So obviously let's start with the baseline. This is a fully connected, deep feed-forward neural network. You see from left to right the propagation of data. So X is a vector, and W's are weights, and computation is that you just multiply those in as a dot product and then apply an activation function, and the result is then H1, 2 and 3, and finally Y as an output vector. And obviously you change only the weights to make this function fit your data. Okay, so make this function fit your data is actually quite interesting, because a neural network, as a reminder here, because of the universal function approximator theorem, a neural network is a universal function approximator. That means it can represent any mathematical function with one limitation, because you have to train it, and you cannot train it in a way that you can represent all the mathematical functions existing out there. So that's the actual limit. So the training is the limit, not the actually predictive power of the neural network itself. Then the whole thing started basically 2015 with the introduction of convolutional neural networks. The idea here is to reduce feature space by bringing things more closely together and preserving, in case of images, the spatial properties. And there are those convolutional layers, and actually the convolutional matrix, which is a filter. That matrix is learned from data, so it's part of the gradient descent training process. Another often occurring architecture pattern is the outer encoder. You see here a neural network, and you have the input and output vectors, and it's X and X prime. They have the same dimensionality or size or whatever you want, and C in the middle is smaller. And what then happens if you train such a neural network, you will recreate X prime from X, and you have to go through the bottleneck C. That means you aren't basically allowed to learn irrelevant things or noise. So there are a couple of applications for autoencoders, for example denoising, or another very important and interesting property is you can use the neural network once trained. So once it is doing a good job in recreating your data, your X prime from X, you can obviously also read out C and use this as a dimensionally compressed vector. And this is a little example of MNIST. If you do that in MNIST, then and you take the dimensionality of C equals 2, then you can plot basically the 768 dimensional space of MNIST into 2 dimensions, and you clearly see that you can easily draw boundaries in 2D space. Then another application of such an autoencoder is embedding things. So here it's word embedding. Word2vec is a famous but old thing. There's obviously a newer stuff going on in that space, but the idea is the same. If you do it for text, you get for each word in the text low dimensional vector representation, and if you project this, then you will see that you have related words being close together in hyperspace. That means you can actually compute with those vectors. So the vec function gives you back the compressed vector for the word. So you get back a vector for the word king, you subtract man, you add the vector for woman, and you get a new vector which is close to the low dimensional representation of queen. So it preserves even the sense. That idea was used to apply to DNA sequence data, and it actually works the same way. The only thing is you use k-meres instead of words, and it works exactly the same way. Another architecture piece or component we have to introduce are Lstms, which stand for long short-term memory. And the idea here is, in all the other architectures we have introduced so far, there is no state preservation, no? And DNA are sequences, so it might be useful if we can somehow preserve state. So let's consider xt to be a vector and t the actual timestamp or sequence index, whatever you want to call it. So in this case, obviously it's a sequence index, but it's also used for time series, so then it's obviously a timestamp or time index. Anyway, what you see here is you have a couple of things going on here. So you have activation function applied to xt, then you have dot product, that x symbol, and the dot product is between the actual activated xt plus the output of the input gate. I will come back to that later, how that is trained. Then this goes into CT, that's the cell state. So each time step then gets represented as a cell state. And from there are many things going on. One is the dot product over the forget gate output. And another one, you see here, there's an output gate. I have a mouse. Oh, sorry, I didn't know that I have a mouse. It's more easy. Okay, so then you see you have the output gate, and that actually after the cell state is activated again, dot product is computed, and then the hidden state is actually output. And here finally you have the forget gate that basically controls how much of the already learned information will be preserved and reused and how much won't. Okay, that's just let's say a 3-sentence summary of the Lstms, and it's actually 3 equations which you need to write down the Lstm cell. So you have different shapes of Lstms. One is a one-to-one mapping. Obviously you can also map a single input to many outputs. It's just how you design a neural network, no? The other way around, you have many examples in a sequence in the input and just map to one output. Or you can do many-to-many, so sequence-to-sequence mappings or subsequence-to-subsequence mapping, and you just slide over it in different types of windows, either tumbling or sliding windows or whatever, or exact one-to-one many-many mapping. That's purely defined in your neural network architecture, how many input and output examples you basically show the neural network at the same time, and obviously then you have to adjust for the input and output dimensions. And then the transformer, that's the latest and greatest, and I won't dig too much into it because I'm just seeing that it's not that much applied in DNA sequence analytics and deep learning, but the idea is basically that you are controlling which parts of the input sequences are more relevant than the others, in a nutshell. And then this is another friendly reminder here that the whole purpose of this intermediate processing steps is to non-linearly transform data in hyperspace. So this here is an example, a deep variant. This is a universal SNP caller, and state of the art is GATK, and you see here the problem in SNP calling is the noisy data. No, you have up to 10% error in your sequencing reads. The errors are complex, systemic errors. And you see here the state of the art in GATK, what is done. So base error modeling using LR, then read likelihood estimation using hidden Markov models, variant identification using naive Bayes, and false positive identification using GMMs, and handcrafted stuff. And there's a lot of manual tweaking necessary depending on your platform. So what's done in deep variant is actually you transform those into 6D tensors. It's more like an image processing pipeline, and you train the classifier with already confirmed and known variants, and basically that hidden knowledge of the true variants, the true biological variants, then how is reflected in the neural network and is used for training. We have some other examples here. So that's NLP inspired algorithm for predicting methylation. It's obviously a supervised labeled data set, and you see here there is some data transformation going on. So you create k-meres, in that case 4 meres, and then you use here the outer encoder, and then you get some sort of a word-to-vec, so embedding for those 4 meres, and then you run a single dimensional convolution. So that's actually a filter which is trained to highlight specific subsets in this vector space, and then you have a flatten and dense layer, and obviously after that you get just a single value, sigmoid something between 0 and 1, and that's actually telling you if that is a methylation site or not, with a probability. Here another example that's interesting because you combine convolutional and recurrent layers here. So you have here first convolution and afterwards recurrent. So convolution for filtering and making sure irrelevant parts are not taken into consideration, and Lstms to just remember the hidden state. Then that's one of the few papers which is using basically a transformer model for actually labeling sequence parts. So it's a supervised model as well. And then here's another one for promoter region prediction. That's also supervised system. So basically it's just always sequence in, and then you get either a probability distribution per nucleotide or per n-mer or k-mer. And here is another one, an example. I'm just showing you a lot of examples because I want you to notice that the parts of the architecture are repeating. No, it starts often with a convolution, and then a fully connected layer, or sometimes there is LSTM involved. Here another one, convolution, pooling convolution, and here there's a GRU involved. The GRU is something similar than the LSTM cell. Just think of it, LSTM cell without the output gate. And here another one. So the thing is, here someone just noticed that it's always some sort of convolution and then some sort of recurrent layers which are performing well, and those folks actually found out and have proven that that's the pattern. No? Convolutional layers and then recurrent layers are always performing best. So they actually created a system for you to just apply to your data and find out which architecture works best. So it's limited to a subset of architectures, but if you remember my first slide, I just added the word automation into it. So that's the current focus I have. It's also outside life science. It's definitely something I'm applying to many other domains as well, but I'm just using a system how to automate easily neural architecture search. And neural architecture search is basically how we do it, nothing else than some sort of hyperparameter tuning. So we actually express the model or the different variants of the architectures as hyperparameters. For example, number of layers, number of units per layer, different type of activation functions, type of layers. So for example, you can just encode the layer type with a number like convolution, recurrent, LSTM, GRU, and so on. And then you can just throw it at a lot of compute. And to make this easy, we are using here Kubeflow, and Kubeflow has a component called Katib. This is a Arabic word which stands for secretary, and that's, in my opinion, the way to go. So all the publications are basically doing nothing else than a little data transformation to fit the data their needs, apply a couple of known architectures, most likely convolutions, and then some sort of recurrent set of layers, and then just manually test them until they get good results. And that's something you can fully automate, and that's why we are here using Katib. And Katib is actually just a wrapper to scale this on Kubernetes. So under the hood there are all the known hyperparameter tuners like HyperOpt and so on running, so you can make use of all those algorithms. So there are a couple of 12 different algorithms which are used in hyperparameter search, and all those are available. So I'm actually doing this anyway. I'm always interested in real problems. So if you have a problem you want to solve with deep learning, just let me know. I'm sure we can just run it through our cluster and maybe come up with a good architecture.